Welcome to our video on the top 5 peptide characterization techniques for accurate analysis. Today, we're diving deep into the methods that help researchers and pharmaceutical scientists ensure the identity, purity, and functionality of peptides. This video is brought to you by ResolveMass Laboratories. If you're working with peptides, whether in research or drug development, understanding these techniques is crucial for ensuring accurate results and reproducibility. Peptides are fascinating yet delicate molecules. They're small enough to be overlooked, but complex enough that even tiny errors in characterization can lead to significant issues downstream. Imagine developing a therapeutic peptide and discovering that batch-to-batch variations are causing inconsistent results. This is where peptide characterization comes in. At ResolveMass Laboratories, we specialize in advanced, validated methods that ensure peptides retain their structural and functional integrity. Today, we'll guide you through the top 5 techniques that are essential for accurate analysis. Before we dive into the techniques, let's understand why characterization is so important. Accurate peptide characterization ensures molecular identity is verified, batches are consistent, and drugs are safe and effective. It's also a regulatory requirement. Agencies like the FDA and EMA demand rigorous analysis before therapeutic peptides can reach patients. Skipping characterization can lead to false results, compromised clinical outcomes, and even regulatory setbacks. So, this isn't just a technical step. It's a vital part of ensuring quality and reliability. Our first technique is mass spectrometry, often called the gold standard for peptide analysis. Think of it as the ultimate molecular scale. MS tells us the precise molecular weight of a peptide, identifies post-translational modifications, and can even help us sequence unknown peptides from scratch. At ResolveMass, we use high-resolution MS systems that provide unmatched sensitivity and specificity. This means even minute modifications or impurities can be detected, ensuring that nothing slips through the cracks. For researchers, this is invaluable when you're trying to confirm that your peptide is exactly what you intended it to be. Next up is HPLC, high-performance liquid chromatography. If MS tells you what your peptide is, HPLC tells you how pure it is. HPLC separates your peptide from impurities, degradation products, or byproducts, giving you a clear picture of your sample's quality. At ResolveMass, we use both reversed phase and ion exchange HPLC methods, which means we can provide both qualitative and quantitative insights with precision. For any peptide development project, HPLC is an essential checkpoint before moving forward. Amino acid analysis, or AAA, is our third technique. It's a bit like doing an inventory check for your peptide's building blocks. AAA measures the composition and quantity of amino acids in a peptide, confirming the structure and making sure nothing is missing or out of place. Using advanced derivatization and detection methods, we ensure absolute accuracy, particularly important for therapeutic peptides, where even a small error could impact efficacy. Think of it as quality control at the molecular level. The fourth technique is NMR spectroscopy. Unlike MS and HPLC, which focus on composition and purity, NMR gives us a window into the 3D structure of peptides. NMR allows us to study how a peptide folds, interacts with other molecules, and behaves in different environments. Understanding these dynamic interactions is key for designing effective therapeutic peptides. At ResolveMass, our scientists interpret NMR data to provide actionable insights, not just raw numbers. This is where peptides go from being just a sequence to being a functional molecule. Finally, circular dichroism, or CD spectroscopy. CD helps us understand the secondary structure of peptides, whether they form alpha helices, beta sheets, or other folding patterns. CD is particularly useful for quick screening and monitoring changes in structure under different conditions. At ResolveMass, we use CD to support fast-paced peptide development workflows, ensuring that structural integrity is maintained throughout research and production. It's like giving your peptide a health checkup for its shape and folding. Let's see these techniques in action. We worked with a mid-sized biotech firm in the US on a novel anti-cancer peptide. The challenge? The peptide degraded rapidly and batches weren't consistent. We implemented an integrated approach. Mass spectrometry detected subtle deamidation. NMR confirmed folding changes affecting stability, and CD mapped secondary structure shifts under varying pH conditions. The results were impressive. Batch stability improved by 42%, impurity levels dropped by 65%, and the client successfully filed their IND application within 6 months. This case highlights how combining multiple techniques gives a complete, actionable picture. So, what's the takeaway? Accurate peptide analysis isn't just about picking one technique. It's about understanding the strengths of each method and using them together. At ResolveMass Laboratories, we bring experience, precision, and trusted expertise to every project. Whether you're validating a therapeutic peptide or exploring new research avenues, choosing the right characterization techniques is critical. If you want to elevate your peptide research, explore our peptide characterization and sequencing services, and let our experts guide you toward accurate, reliable results. Thanks for watching. Don't forget to like, subscribe, and hit the bell icon for more insights into peptide research and analytical techniques.